This is an IGCSE Edexcel exam paper talk through from January 2021. The diagram shows a plant cell. Which part of this cell contains chlorophyll? So you're looking for the chloroplast, which is S. Which of these is found in chlorophyll? Okay, they want you to know your ions very well. Calcium, remember, is all to do with strong teeth and bones, so irrelevant with the plant. Iron is about healthy blood. It's actually magnesium. You need magnesium ions to make the chlorophyll. Describe the role of chlorophyll. Chlorophyll traps light and carries out photosynthesis, that number one plant process. Because what's the whole purpose of that? To make glucose. Which of these is an example of positive phototropism? So photo means to do with light. Positive means growing towards the light. So a plant root growing away from the light, no, because it's away. A plant root growing downwards towards gravity, no, because that would be positive geotropism. A plant stem growing towards light, yeah, look, there's the light, growing towards. So it's C. A plant stem growing upwards due to gravity, no, that would be negative geotropism. The table lists the roles of some substances found in living organisms. Complete the table by naming each substance. The first one has been done for you. Digest fats. You need an enzyme, that is lipase. Diffuses across the synapse. Remember, that's a gap between two neurons. It's a neurotransmitter. Prevents scurvy. Gosh, this is so niche. That is vitamin C. The diagram shows the position of some hormone-producing glands in the female body. Which of these structures is the adrenal gland? Now, renal means to do with the kidney. Adrenal means next to the kidney. We can see the kidneys here, which is why Y is the answer. The adrenal gland is an organ that secretes adrenaline. State what is meant by the term organ. A group, learn these definitions, a group of similar tissues, the key word here is tissues, working together to perform a function. Adrenaline is released into the blood when there is danger. The list gives the effects of adrenaline on different parts of the body. It dilates the pupil in the eye. It increases heart rate. It narrows the small arteries in the intestine. It converts glycogen into glucose in the liver. Explain the advantages of these effects to the person in danger. So why is pupil dilation good? Well, it allows more light to enter the eye. And why is that good? So you can spot danger. Why is increasing the heart rate good? Well, that will deliver more oxygenated blood to tissues and muscles. And why is that good? To allow them to respire more and therefore contract. And muscle contraction is important so that you can run away from the danger. This narrowing of the small arteries in the intestine means that blood is diverted to the muscles, which again ties into our previous point. And that means some more oxygen and glucose is delivered. And then lastly, converts glycogen to glucose in the liver. We've already kind of talked about the glucose there, and I know I've got plenty of marks. I'll just show you where they're awarded. More light to enter the eye is 1 mark, so you can see danger is 2 marks. More oxygen to the muscles, 3 marks. They can respire more, 4 marks. Muscle contraction, 5 marks. And glucose being diverted to the legs, 6 points. The diagram shows part of the human digestive system. In which of these parts is hydrochloric acid produced? You're looking for the stomach, which is B. In which of these parts are feces stored? So that's poop. You're looking for the rectum, that's D. The liver produces bile. Explain the role of bile in digestion. So, Bile emulsifies fats. That's a good word to use here, meaning large fat droplets are broken down into small fat droplets. Why is that a good thing? Because it increases the surface area for lipase to act on. So effectively the bile takes those fats and turns it into these small fat droplets which have a much larger surface area. It has a second role, which is that bile neutralises hydrochloric acid from the stomach, therefore providing, use nice keywords here, optimum pH for enzymes in the small intestine. Some people have a condition called celiac disease. In this condition, the body reacts to eating gluten, a protein found in wheat. This reaction damages the villi in the small intestine. The diagram shows how the villi in the small intestine are damaged. Explain how the undamaged villi are adapted for their function. So let's zoom in on this. What can we see? Well, these finger-like projections will increase the surface area. You can't see them, but they'll also have microvilli on the top, which increase the surface area more. Inside we can see the blood capillaries, which will absorb that digested food. You have a thin wall here to provide a short diffusion distance for those digested food molecules. And then lastly in the middle you have what's called a lacteal, which will allow fat absorption. Explain why children with untreated celiac disease may grow more slowly and become tired more easily than children without celiac disease. Use information from the diagram and your own knowledge to support your answer. So if we have a look, we can see that the villi are a lot shorter. This means that you're going to have a reduced surface area, meaning that you have less absorption of digested food. What sort of food could that be? Well, that is things like glucose, which means they'll be able to carry out less respiration. They'll potentially absorb fewer amino acids, so they'll be able to build fewer proteins. They'll have less vitamins. They might suffer from something like scurvy. If it's a lack of vitamin C, a lack of vitamin D means they could have rickets. They may absorb less iron, which remember is needed to prevent anemia. And that anemia is the disease where they get tired more easily. So you can see how we're building up a nice full answer here. A teacher does an investigation to show that plants require carbon dioxide and light for photosynthesis. This is the teacher's method. Place a potted plant in the dark for 24 hours. Place a strip of black paper over 2 of the plant's leaves. Pour some sodium hydroxide solution into a flask. Insert one of the leaves into the flask. Seal the flask with a cotton wool plug. Place the plant in bright light for 12 hours. Remove the 2 leaves and safely test them for starch. Explain why the potted plant is placed in the dark for 24 hours. That's actually to stop it photosynthesizing. And so because of that, it can't make any glucose and it's forced to use up its starch. Explain one role of leaf Y in the investigation. It's going to act as a control because if you think about what's going on here, the sodium hydroxide will absorb all the carbon dioxide. So the leaf acting as a control will show that light is needed for photosynthesis as well as carbon dioxide. Describe how to test the leaves for starch safely. There's a key word. So you need to boil in ethanol, remember, to remove chlorophyll, because that green of the chlorophyll will obstruct the iodine which we need to test for the starch, but use a water bath. There's the safety aspect. Now add the all-important iodine solution, which should turn blue-black if starch is present. And add another safety feature, wear safety goggles. Explain how the results of this investigation would show that light is required for photosynthesis. So we need to consider what would be happening with leaf Y. Here, because that black strip of paper is on the leaf, then no photosynthesis would be taking place, meaning that there'd be no starch present, so you'd expect it to stay orange, whereas the rest of the leaf would return blue-black. Plants convert the glucose they produce into starch. Explain why plants store carbohydrate as a starch rather than glucose. This is an answer that's all to do with osmosis, because starch is insoluble and therefore is a great storage compound as it has no osmotic effect. So it won't be drawing water into and out of cells. The chromosomes found in human cells can be photographed and arranged in order to produce a karyotype. The karyotype shown in diagram 1 is from a normal human body cell. Let's have a look down here. Because we have that XY, we have a boy. Explain how you can identify the sex of the person from the karyotype shown in diagram 1. So I'm sure there's a mark for saying it's a boy because they have the XY sex chromosomes. Remember, if it was a girl, they'd have 2 X chromosomes. Cells can be described using the terms diploid or haploid. Explain the difference between these two terms using the information in diagram 1. Let's have a look at what we've got going on here. We have diploid because remember, diploid means contains 2 sets of chromosomes. If it was a haploid diagram, there'd only be 1 set of chromosomes. The karyotype shown in diagram 2 is from a body cell of a person with a condition called Klinefelter syndrome. This condition only affects males. Describe the differences between the karyotype in diagram 1 and the karyotype in diagram 2. Let's have a look down here. So you had the XY before and there were just 2 chromosomes. So the sex chromosomes here, we can see that we have an extra chromosome. And if you have a look, that is the X chromosome here, the longer one. So we can see we've got 2 X chromosomes here and 1 Y chromosome. So diagram 2 has an extra chromosome. Let's point out specifically an extra X chromosome. Suggest how the karyotype in diagram 2 may have been caused. Let's think about how we could get that extra chromosome. Well, remember in meiosis, the chromosomes have to separate. So potentially if the chromosomes didn't separate properly during meiosis, you would find that you'd gain that extra chromosome. The frequency of Klinefelter syndrome in the United Kingdom is 1 in every 660 males. Oh my gosh, that is so many. The population of the United Kingdom is 66 million, of which 49% are male. Calculate the total number of males in the United Kingdom with Klinefelter syndrome. Let's work out how many males there are then. So we need 49% of 66 million. And then finally divide that number by 660 to work out how many Klinefelter males we'd have. So it's 49,000. Suggest why females who are aged over 35 are more likely to give birth to a baby with Klinefelter karyotype, simply because the female's eggs, the gametes, are older and therefore more susceptible to the sort of mutations which would lead to the ineffective separation of the chromosomes. Scientists have developed genetically modified GM crops in order to increase food production by increasing crop yields. Some GM crops are described as transgenic. Explain what is meant by the term transgenic. It means containing a gene from, has to be a different species. Give the role of 2 named enzymes in the production of GM organisms. I want to talk really about the ligase enzyme and the restriction enzyme. Remember, the ligase joins DNA to the bacterial plasmid whereas the restriction enzyme cuts the DNA. Some GM crops that are available to farmers are resistant to herbicides, weed killers. Some GM crops are resistant to diseases caused by viruses and to damage by insects. So from a crop productivity point of view, these are both good things. Some people are for the use of GM crops because they may be beneficial to ecosystems. Some people are against the use of GM crops because they could harm ecosystems. Discuss these opinions for and against the use of GM crops. 4 marks. So looking at points for the use of GM crops then, it would mean that less pesticide is used, and that's a good thing because it means you have less bioaccumulation of pesticides. Because the GM crops are resistant to herbicides, to the weed killers, it means that when those weed killers are applied, it will just be the weeds being killed, and therefore they'll be easier to remove. But what about the disadvantages of using GM crops? We know that they could be harmful to ecosystems, so what sort of ecosystems? Well, what about these insects? You definitely see a reduction in insect population, and when numbers of species populations vary, that means you get a disruption of a food chain, which is an adverse effect on the ecosystem. In some countries, snails are farmed as sources of protein. The photograph shows a snail. The scientist investigates the effect of temperature on the growth of snails. The scientist measures the mean average shell height of the groups of snails kept at 3 different temperatures for 24 weeks. Plot a graph to show how the mean shell height increases with time for each temperature, and use a ruler to join the points with a straight line. Let's just have the data here so we can see it. So what we're measuring goes on the y-axis, and that is the mean shell height in millimeters. Versus our x-axis, which is time in weeks. So pick a sensible scale here, occupying as much of the graph paper as possible. We go from 15 millimeters all the way up to 21.8. So let's shrink that scale a bit. So let's start with the 8-degree one. So at time 0 weeks, we have a 15 mil shell. Then at 8 weeks, we have 15.2. 16 weeks, 15.5. 24 weeks, 16.4. Use a straight line to join your points. And then we need a key saying that that was for 8 degrees. What about now at 15 degrees? And lastly at 23 degrees. Cool, so that's done. Explain the effect of temperature on the growth of snails in this investigation. So why does a higher temperature mean that the snails grow faster? Higher temperatures, the enzymes involved in all the chemical reactions that take place inside the snail have more kinetic energy, meaning that there are more collisions between those enzymes and their substrate molecules, so more reactions will be catalyzed, and therefore as a result we can say that increased temperature has therefore increased snail growth, which we can see, look at the growth of the snail at the 23 degrees mark versus 15. State the dependent variable of this investigation. That's what you're measuring. It's the snail shell height. State how the scientists made sure their results were reliable. That's all to do with repeating. They repeated their experiments. Assimilation efficiency is the percentage of food that is eaten and not ingested as feces. Assimilation efficiency is calculated using this formula. A snail eats 1.2 grams of food and produces 0.3 grams of feces. Calculate the assimilation efficiency of the snail. So we'll take this equation. So the snail ate 1.2 grams of food, produced 0.3 grams of feces. Let's divide it by that mass of food eaten, which was 1.2, and then times it by 100. So it becomes 0.9 over 1.2 multiplied by 100, which comes out at 75%. Explain why the assimilation efficiency of a primary consumer is less than the assimilation efficiency of a secondary consumer. So why would the primary consumer be obtaining effectively less energy compared with the secondary one? Well, what's it eating? It's eating plants, producers, whereas the secondary consumer is eating animals. So what's true about plants? Well, they've got an awful lot of indigestible matter, and therefore more feces will be made, hence more wastage. The production efficiency of an animal is the percentage of assimilated food that is made into new biomass. The table shows the production efficiency of a mammal and a snail, both of which are primary consumers. Suggests why there is a difference in the production efficiency of the mammal and snail. So what's true about the mammal? Well, they're warm-blooded, which means they need to maintain a steady temperature, which is high. They also experience heat loss to the surroundings, so that's going to be where a lot of this is wasted. The diagram shows an insect transferring pollen grains from flower P to flower Q. Which structures in flowers contains pollen grains? It's the male part, remember, it's the anther. Pollen grains are deposited on the stigma and grow tubes down to the style. Suggest how style tissue helps the tube to grow. Now the key word here is the fact that the tube needs to grow, so that means elongation, and in order to do that it needs things like carbohydrates, it needs amino acids. So we're going to say that the style tissue provides glucose needed for respiration and amino acids to make proteins. The graph shows the change in length of a pollen tube over a 180-minute period. Calculate the fastest rate of the pollen tube growth in millimetres per minute. 3 marks. So the fastest is the steepest portion of our graph, which is here. In order to find the rate, we have to do the gradient. So our change in y here is 4.7. Our change in x is 60. So it's grown 4.7 millimetres in 60 minutes. So actually that will give us our growth in millimetres per minute if we just pop that into our calculator to get a value of 0.055. A farmer grows apple trees. The flowers on the apple trees must be pollinated by insects to produce an apple crop. The farmer is concerned that the use of pesticides may affect the yield of apples. Design an investigation to find out if pesticides reduce the yield of apples. Include experimental details and write in full sentences. So remember, if you've been following me for a while, I like to use the variable layout here rather than columns. So what will our independent variable be? What will we change? The independent variable is the volume of pesticide applied. It needs to be something tangible. What's our dependent variable? So what we're measuring, it's the yield of apples. But again, what will we actually measure? The number of apples. We want at least 3 control variables. So what are we keeping the same? So what will affect that growth of the apple, the yield of the apple? The temperature, the light intensity, carbon dioxide concentration, growing period, variety of apple. This is a great question. There's so many control variables. I've gone crazy. I haven't just included 3. And then to get your final marks, you need to state a time period. So carry out for 6 months. Pick something sensible, repeat, and calculate an average. And by doing this, you'll make sure you get those 6 marks every time. Aerobic respiration uses oxygen and produces ATP. Complete the word equation for aerobic respiration. So we need oxygen plus glucose produces carbon dioxide and water. A scientist investigates the rates of aerobic respiration of some animals. The scientist calculates the rate of respiration per gram of each animal. The results are shown. Explain why a mouse uses more oxygen per gram than a human. Wow, look at these numbers. Okay, what do we know about a mouse? Well, firstly, the mouse is much smaller and therefore has a much larger surface area to volume ratio. So because of that, remember that both the human and mouse are mammals and they're going to be losing heat to the surroundings. Therefore, because the mouse has a much larger surface area to volume ratio, it needs to carry out much more respiration, therefore use up much more oxygen in order to maintain its body temperature. The structure of a frog heart is different to the structure of a human heart. The diagram shows a section of frog heart with arrows showing the direction of blood flow. What is the name of the blood vessel labelled X? Okay, it's coming into the heart, which remember means it must be a vein. It's coming from the lungs. The word pulmonary means relating to the lungs, so it is the pulmonary vein. C. Give one difference between the structure of the frog heart and the human heart. And just see, really, there's way fewer chambers. So the human has 4 chambers, whereas, have a look, the frog has 3 chambers. You could also mention the fact that there's no septum in the frog heart. Remember, that's the division between the left and right side, or the fact that it only has 1 ventricle. Humans can run for long periods of time, whereas frogs can only move for short periods of time. Explain how the structure of the heart of a frog means it is unable to move for long periods of time. 5 marks. Woo! No septum means that there's no separation between the left and right side of the heart. So what does that mean? Well, it means that oxygenated and deoxygenated blood mix. And that's not good because it means that less oxygen reaches respiring tissues. So less ATP will be made. Use the information they've given you higher up in the question. And that's very important for that frog muscle contraction. Let's look again more closely at that heart. We can see that there are no semilunar valves, and they're very important for preventing the backflow of blood, as well as no atrioventricular valves. Again, you'll end up with backflow back into the ventricle. So these are all things which mean that the frog is not very good at efficiently delivering oxygen around the body. I'll show you where the 5 marks are awarded. We've got the oxygenated and deoxygenated blood mixing, less oxygen reaching respiring tissues, less AT being made, no semilunar valves, more backflow of blood. So there's your 5 marking points here. Blood consists of cells and plasma. Plasma transports various substances to and from different parts of the body. Describe the function of plasma in transporting named substances in the body. Okay, this is quite a run of difficult questions. So what does the plasma transport? Well, it contains glucose, amino acids, fatty acids, hormones, urea, carbon dioxide, antibodies. But make sure you answer this from the right angle because it's saying describe the function of plasma in transporting named substances in the body. So really it's saying where are those substances coming from and going to? So plasma transports glucose from the small intestine to respiring body cells, fatty acids from the small intestine to respiring cells, hormones, e.g. insulin from the pancreas to the liver, carbon dioxide from respiring cells to the lungs. The diagram shows a white blood cell called a phagocyte. The magnification of the cell is calculated using this formula. The actual diameter of the cell is 0.013 millimeters. Calculate the magnification of the cell. So you want to measure the diameter of this phagocyte. Make sure you use your ruler and you measure it in millimeters. So that becomes 58 millimeters. It's so important that these units are the same. And then do 58 divided by 0.013, and your magnification is times 4,460. Name the structure labelled A in the diagram. It's quite an unusual shape because it's lobed, but it is a nucleus. Describe the function of this cell in defending the body from infection. So a phagocyte engulfs pathogens and digests them using enzymes. Other white blood cells produce proteins called antibodies. State how you could test a sample of plasma for protein. Oh wow, we've just suddenly dived into food tests, so use Biuret reagent, which should turn purple in the presence of protein.